Dear students, this is the first lecture on Chapter 5, primarily dealing with DNA structure and some chromosome function. In Chapter 2, we learned some information about biomolecules, and that information will be relevant as we move forward in Chapter 5. We know that nucleotides are the building blocks of DNA. We need to learn now that nucleosides are the building blocks of nucleotides, and we'll see that shortly. We need to know the major difference between DNA molecules and RNA molecules from the structural perspective. In this chapter, the student is required to learn this information in much more detail. One startling fact to emerge from all our research is just the understanding of the amount of DNA packaged into a single nucleus. We learned in Chapter 2 that DNA is a macromolecule. But just how big this molecule is illustrated in this chapter. It has been calculated that if the DNA from a single nucleus was extracted and unwound so that just the DNA helix existed, the total length of all the DNA fragments would be over 6 feet in length. This DNA is packaged not into a single cell, but into the actual nucleus within that cell, the nucleus being just a few micrometers in diameter. The question then is, how has nature not only squeezed in this enormous amount of DNA into a tight space, but also how that DNA is regulated? Incidentally, the number 24 in this particular fact pertains to one copy of each type of chromosome present in the human species. So that will be the 22 of autosomes plus the 2 sex chromosomes, the X and the Y. DNA is short for deoxyribonucleic acid. DNA is an acid because of the phosphate groups, which become highly polarized when DNA is dissolved in water. The word macromolecule really does apply to DNA. The individual building blocks of DNA, the monomers, are called nucleotides. All DNA double helix molecules have a directionality for each of the strands. We'll cover this a little later with some diagrams. Both DNA and RNA molecules can be extremely large. Both DNA and RNA are constructed of nucleotides as the building blocks. Nucleotides are linked together through special types of covalent bonds to form single strands. These single strands may, in both cases, form a double helical structure, especially for DNA, which is known as the double helix. RNA molecules can temporarily form double-stranded regions, as we'll see later. The double helix itself is managed and nursed by accessory proteins whose job it is to bind and twist the DNA, forming its three-dimensional structure found along chromosomes. This slide recapitulates the information presented in the previous slide, whereby the building blocks of DNA and RNA are nucleotides. In the case of DNA, double helical structures may form, which are then twisted and turned to form individual chromosomes. Nucleotides themselves are composed of chemical subunits. There are 3 essential components to a nucleotide: a phosphate group, a sugar, and a base. Some of these components are common to both RNA and DNA, while others are unique. For instance, both RNA and DNA molecules contain the same exact phosphate groups. However, the sugars do differ between RNA and DNA. DNA is always composed of deoxyribose. With respect to the bases, DNA has purines and pyrimidines. The purines found in DNA are adenine and guanine, and the pyrimidines found in DNA are thymine and cytosine. Rarely, if ever, is uracil found within a DNA molecule. With respect to RNA nucleotides, the sugar is ribose, and instead of thymine, uracil is used as that nucleotide base. A useful learning aid that helps me in understanding the relationship between the bases is the term pure as gold. Pure codes for purines. As reminds me that adenine is a purine. Gold further reminds me that guanine is also a purine. The use of gold is a further reflection on the heaviness of this particular metal. Gold is heavy, so are double-ringed bases. So adenine and guanine must be purines, and purines are composed of 2 rings, not 1. The physical arrangement of the 3 components is indicated in this particular slide. For DNA, the important thing is that the sugar in between the base and the phosphate in the nucleotide is deoxyribose, and that is indicated by the second carbon on the sugar ring having no oxygen, thus deoxy. Whereas in RNA molecules, RNA nucleotides, the oxygen is present on the second carbon on the sugar ring. This makes the sugar ribose. Additionally, this slide indicates which atoms are involved in connecting one nucleotide to its neighbor. This oxygen atom on the phosphate is important in connecting the nucleotide above, whereas this hydrogen on the sugar is important in connecting to the nucleotide below. The phosphate of the nucleotide below will be bonded directly to that oxygen. Likewise, in RNA molecules, the same atoms are utilized in the formation of nucleotide polymers. So, in summary, a base plus a sugar is called a nucleoside. Examples of nucleosides will be adenine plus ribose and adenine plus deoxyribose. Once you add the phosphate, then the nucleoside becomes a nucleotide, the building blocks of nucleic acids. In this case, adenosine monophosphate would be AMP, adenosine diphosphate would be ADP, and adenosine triphosphate would be ATP. These molecules can also be sequestered for other purposes within the cell. As we'll see, the polymerization of nucleotides with neighbors begins the formation of a polymer. In this particular case, one is beginning to see how different atoms on the sugar are orientated. So the third carbon on the sugar is facing south, whereas the 5 carbon on the sugar is facing northwest. This orientation is very important. Examining the structure in depth shows us how the sugar phosphate backbone is connected between neighboring nucleotides. So the bottom nucleotide here is connected to 3 further nucleotides above. Here the phosphate group of the bottom nucleotide is connected directly to the 3rd carbon of the nucleotide above. And this pattern repeats itself with the remaining nucleotides. These bonds are known as phosphodiester bonds. Diester because of the esterification of the oxygens to the carbons. Di means there's 2 of them on either side of the phosphate, so a phosphodiester bond exists between nucleotides. These are covalent bonds holding the backbone in a very stable configuration. The other note is the arrow indicating the direction of the backbone, with the 5' end labeled at the top, the 3' end labeled at the bottom. This is in reference to the carbon atom on the sugar, which has the very terminal phosphate group. Here the final structure is a phosphate group, and this end of the molecule is thus labeled 5'. At the other end of the same molecule, we do not have a phosphate group, but we do have the free hydroxyl group on the 3rd carbon of the sugar molecule. Thus, this end of the DNA strand is labeled 3', 5' and 3'. As mentioned several slides prior, nucleotides can come in various different flavors. In this particular case, we're looking at the base adenine bonded to a ribose. This forms the basis of other molecules within the cell that have important functions. ATP is considered the rechargeable battery within cells. Millions of ATP molecules are converted almost instantly between ADP and ATP. The difference between ATP and ADP is indicated by these brackets. ATP has 3 phosphate groups linked together in a chain. ADP would have just 2 phosphate groups. The third phosphate group is removed, and in the process, this bond here is cleaved, and that releases a lot of energy that can then be utilized to drive other chemical reactions within the cell. Further reiterated in this slide is the connection between the carbons of the sugar and the 2 neighboring entities. Bases are always connected to the first carbon of the sugar molecule, and phosphate groups are always connected to the 5th carbon of the sugar molecule. To summarize the general patterns shown above in the figures, we see that individual nucleotides are bonded together with their neighbors through covalent phosphodiester bonds. A phosphate connects the 5' carbon of one nucleotide to the 3' carbon of another. The strand therefore has directionality. One end of the molecule will always end as a 5' free phosphate group. The other end of the molecule shall always end with a 3' hydroxyl group. Neighboring phosphates and sugar molecules form what is known as the backbone of the nucleic acid strand. This is true for RNA and for DNA. Summarizing the information pertaining to DNA, the full name of DNA is deoxyribonucleic acid. It is a macromolecule. The subunits of this macromolecule are called nucleotides. The backbone has an orientation. It runs from 5' to 3'. Those 2 strands, when they come together, are then orientated in opposite directions, as indicated by this slide here. So in the configuration normally DNA is found in inside cells, we have a double spiral helix. In this cartoon, we can unwind the DNA to illustrate the relationship between the 2 strands. On the left, we have the molecule that runs in one orientation from 5' to 3', and the only way the other strand can make contact with the basis is to run in the opposite direction. So from 3' down to 5'. Another important feature of DNA double helix is that the 2 spirals do not run equal distance from each other. As indicated here, the spacing results formation of a wide gap or a narrow gap between the 2 strands. The wide gap is known as the major groove. The major groove runs in a spiral direction along the DNA, so does the minor groove. This slide presents several important definitions used throughout this chapter and subsequent chapters. The genome is a reference to the complete set of information in an organism's DNA. This would include nuclear DNA plus extranuclear DNA such as mitochondria in the case of animals, and chloroplasts would also be included in the case of plants. Genomes are normally compared based on the haploid complement for the nucleus. Chromosomes, by definition, are really a single continuous molecule of double-stranded DNA. In some organisms, like bacteria, these molecules are circular, so chromosomes are circular. In eukaryotes, in most cases, the chromosomes are linear, each with 2 terminal ends. The definition of a gene is pretty malleable. In the case of our textbook, it's referred to as a region of the DNA coding for the production of a single polypeptide. We saw in chapter 4 that a polypeptide may form the basis of proteins. However, recent research has shown that genes do not have to make polypeptides. They can stop at the RNA level. We'll cover this in subsequent lectures. And a karyotype is defined as a figure or photograph showing the full set of chromosomes taken from a single cell, normally laid out in an orderly fashion from the largest chromosomes down to the smallest. With reference to human DNA, the human genome has been calculated to be approximately 3.2 billion base pairs in length, and this is dispersed over 24 distinct chromosomes. Many authors point out that it would take nearly 10 years for someone speaking at a normal speed to read out the 3.2 billion base pairs of DNA. Since genome sizes are stated for haploid content, it's interesting to know that for most cells in our bodies, we have 2 copies of each chromosome set. So the actual amount of DNA that has to be maintained inside the nucleus of a typical cell is 6.4 billion base pairs, not 3.2, which will be applicable to gametes like the sperm and the egg. It is humbling to understand that humans do not have the most chromosomes of living organisms. Plants have that record. Neither do humans have the largest genome size. Fish tend to have that. Interestingly, a newly discovered ant species in South America a few years ago has just 1 pair of chromosomes. As the next slide will show, there is no real association between genome size and evolutionary complexity. Humans are somewhere near the middle. So on the bottom scale, we have genome size in kilobase pairs, and then we have on the y-axis a listing of creatures from the smallest to some of the largest in terms of genome size. And as one can see, plants vary enormously in the amount of genetic material they possess in different species. Since there is no direct correlation between evolutionary order and the size of the genome, this is known as the C-value paradox. We are beginning to understand the reasons why it exists, but still it's a paradox. The chromosomes of all living creatures are complexed with proteins. It's the protein component which gives chromosomes their shape and structure. During the cell cycle, chromosomes can change visibly in their thickness and their length. During interphase of the cell cycle, the chromosomes are basically invisible under the light microscope, being very thin and very long. During metaphase, the distinct chromosomes can be seen under the light microscope because they have coiled further, thickened, and shortened. The ends of linear chromosomes are known as telomeres. These are important structural regions which protect the chromosome from environmental degradation. The middles of the chromosomes have special regions which help the chromosomes navigate during cell division, and these regions are called centromeres. Revisiting chapter 1, where we learned about the internal structure of cells, the nucleolus is a hotspot within the nucleus where different chromosomal segments containing similar genes are brought together and undergo rapid transcription. These chromosomal arms contain the genes for making ribosomes. A healthy average cell can produce up to 10,000 ribosomes per minute. Therefore, there is a heavy demand on these genes, and nature has duplicated the genes and placed them on many different chromosomes. So the nucleolus is basically an area of active transcription of ribosomal genes. The term chromatin refers to not just DNA or the housekeeping proteins surrounding the DNA, but to the complex of both together. Chromosomes are rarely without their associated proteins. The cell cycle illustrates how dynamic the chromatin structure of cells can be to accommodate different biochemical feats the cell has to perform in order to survive. Historically, DNA was not the favored molecule for heredity. That went to proteins until proteins were disproven through various experiments not to be the molecule of inheritance. Most of the research was performed in the 1940s, as illustrated in the brown pages in our textbook. One of the most important experiments of the time was conducted using different strains of bacteria. The first strain, known as the S strain, S for smooth, because their outer coat was smooth and slippery, was the virulent strain that caused disease in mice. The other strain, known as the rough strain, was unable to cause disease because the surface of those cells was easily identified by the immune system and phagocytosed, i.e. eaten by white blood cells. In a classic experiment, the researchers took dead S strain bacteria and placed them into the same container as living R strain bacteria. After a number of minutes, a small amount of this solution was injected into mice. The mouse subsequently died, and analysis of the blood of the mouse revealed that indeed the strain alive inside the mouse was the S strain. It's impossible for dead cells to become living. So what had happened? It appears that something was passing from the medium containing the dead S strain to transform the R strain into S strain. What was that substance? Was it RNA? Was it protein? Was it DNA? Was it lipid? Or was it sugar? The results revealed that only DNA, when allowed to survive in the solution, resulted in the production of S strain. This classic experiment proved for the first time that DNA was the molecule of inheritance.